we are going to discuss about DNA replication in our course of Molecular Biology 1. So, the structure of double helical structure of DNA was proposed by Watson and Crick in 1953. And when they proposed this structure of DNA to be double helical, using the evidences and information from Chargaff's rule, from the X-ray crystallographic studies, they had also predicted, they had also postulated that because of the complementary nature of DNA, because the two strands are complementary, there might be a mechanism for copying the genetic material. That is, within the structure of DNA lies how the genetic material will be copied or how DNA will be replicated. This Watson and Crick had proposed in their article in Nature in 1953 itself. But scientists were still trying to find out, using different experiments, how would DNA be replicated. And this is the original diagram which Watson and Crick had first drawn as to how the structure of DNA or how DNA would look like. So, from then, many scientists were trying, many experimental evidence were put up to find out the structure of DNA is double helical, but how is DNA replicating itself, or how is DNA duplicating itself, or how is DNA synthesized inside the cell? For that, three models for DNA replication were proposed. So if this is our DNA strand, as DNA is double helix, we have two strands, and these two strands are anti-parallel in nature. Means one strand is running in the 5' to 3' direction and other is running in the 3' to 5' direction. This is what Watson and Crick had proposed. Now, if it is conservative mode of DNA replication, means if DNA is replicating by the conservative mode, we will have both the parental strand retained as it is, and we will get new daughter strand. So that is what will happen in the first generation. Means if a cell has this copy of DNA, after conservative mode, it will get one strand which is having, or one DNA molecule which is having completely the two old strands, and two new strands will be synthesized in the daughter cell. After second generation, one will be the old strand, means one DNA molecule consisting of two old strands and three new DNA molecules. All are having new strands in them. This is conservative mode of replication. These three models were proposed. What were they? Conservative, semi-conservative, and dispersive model. So conservative states that the parental strand will completely stay as it is and a new daughter strand will be synthesized. So at first generation, you will have an old strand and new strand, and after second generation, when there will be four strands of four new DNA molecules, out of that three will be new and one will be the old strand. Now semi-conservative replication. Semi-conservative replication means half of the parental strand will be retained and half will be new. So if we see, this is our parental strand. After replication, we will have two daughter DNA molecules. In that, one strand will be old and other strand will be new strand. After second generation, same thing is happening. So you will have four DNA molecules. Two will be hybrid because one would be from the old DNA, parental one, and one is new. Then this splits up, blue one will come in one parent, another blue one will go in another daughter strand, and in this way you will have four strands in which two are the new ones and two are hybrid. This is semi-conservative, means one parental strand is retained. That is one old strand and one new strand in a DNA molecule of two strands. One is old, that is one is parental, and one is new strand. That is semi-conservative mode of replication. And the last one is the dispersive mode. In dispersive mode, what they are saying is the entire parental strand is broken and new strand is synthesized on it in pieces. So here, after first generation, both the strands will be hybrid. After second generation, again you will get hybrid strands. That is mixture of both old and new. This is dispersive mode. That is the entire DNA molecule is getting dispersed and then all the fragments are combining. So these three modes of replication were proposed. So to prove one mode of replication is followed in organisms, what we need to do is we need to provide an experimental evidence for the same. So experimental evidence was given by Matthew Messeloson and Franklin Sthal. So this experiment is popularly known as Messeloson and Sthal experiment, and they were working on E. coli. E. coli is a bacteria. They were working in the California Institute of Technology where they used two isotopes of nitrogen, N15 and N14. Why? Because 17% of the mass of DNA is nitrogen. Because DNA consists of nucleotides and nucleotides are having nitrogenous bases in them. So if we see the mass of a DNA molecule, we have 17% contributed by nitrogen. So to distinguish between two strands, because here what is important thing in an experiment is we should be able to distinguish between the parental strand and the daughter strand, or old strand and the new strand. Old strand and the new one. Then only we can prove that DNA replicates by a specific mode of replication. So for that they used N15 and N14 isotopes of nitrogen. And the method they used to separate, first thing is to distinguish between the two strands, and second thing is also to separate between the two strands. They use density gradient centrifugation. Because if we have a N15 nitrogen containing DNA, that is heavy isotope containing DNA, there is a difference in mass between N15 DNA and N14 DNA. The difference is only of 1.2 to 1.3%, but using the density gradient centrifugation, we can very easily distinguish between these two strands. So now what they did is, they used E. coli. Okay, this E. coli was first grown in a medium. This medium was containing N15 nitrogen, that is heavy isotope of nitrogen. For few generations, this E. coli was grown on N15 containing medium. So whatever DNA or whatever genetic material this E. coli had, it was having N15 nitrogen in the DNA. And later on, the same E. coli was grown on N14, that is normal or lightweight isotope of nitrogen was grown on this media. And after specific intervals of time, the solution was taken and it was centrifuged. Okay, so 20 minutes specifically, to be more specific, because the E. coli duplicates or replicates after 20 minutes. That is, from one cell of E. coli, two cells are formed after 20 minutes. So this was cesium chloride was used to prepare density gradient. Cesium chloride density gradient centrifugation was performed. Cesium chloride is a salt which forms a specific density in the centrifuge tube, and wherever density of the DNA matches, it will go and settle down over there. Now, as this is N15 nitrogen containing DNA, which they initially used, this one, so its density is high, so it will settle at the bottom of the centrifuge tube in CSCl gradient, that is cesium chloride gradient. After first round of replication, okay, we have intermediate density. Neither it is heavy nor it is light. It is in between. So it is forming a intermediate band. And after second round of replication, there are two bands, one intermediate and one light. So once again, the gist of what exactly Messeloson and Sthal did. They used E. coli cells. This is the genetic material of E. coli, DNA of E. coli. First E. coli was grown on N15 medium and E. coli was grown on N14 medium. Initially, E. coli was grown in N15 medium, and after that the same E. coli was transferred to N14 medium. Then what they did is, after specific intervals of times, cesium chloride density gradient centrifugation was done. That is, DNA was extracted and it was centrifuged in the cesium chloride density gradient form of different densities of cesium chloride molecule. So after first round of replication, all the molecules will be N15. So 100% the band is formed at the bottom. After first round of replication, what is happening is, if it is semi-conservative mode of replication, one will be from the parental strand and one will be the newly synthesized strand. So it will show a intermediate density, means half of the molecules will show a heavy band and half will show the light band. So we will see that over here in detail. This is the control experiment. Control experiment means where is the band of N15 formed and where is the band of N14 formed. So heavy nitrogen will form band at the bottom of the cesium chloride gradient and light band will be formed above it, because it is a light isotope, light in weight. The mass is less as compared to the heavy. So, what results they obtained? What were their observations? Accordingly, they interpreted the result. What they observed is, when E. coli DNA is taken and it is centrifuged, after first round of replication, you get only one band, which is intermediate, indicating there is hybrid formation. Hybrid will be formed, it will be intermediate only when it is semi-conservative mode, because one DNA will come from the parent and one DNA is newly synthesized in it.